Hi, good to see you again. Let us begin our next uh, lecture. I think we have finished two lectures on mitochondrial toxicity. This is maybe the third lecture on the effect of drugs, xenobiotics on mitochondria. We were considering mitochondria as a supplier of energy to the cell. We were considering mitochondria as a powerhouse of the cell. But now, what we are going to discuss, this mitochondria is not only a powerhouse of the cell, mitochondria maintains the cell viability. Mitochondria helps in maintaining the cellular health, and that's very important because you see, the cell is a coordinated structure where we find a coordination between nucleus and mitochondria, between mitochondria and ER, endoplasmic reticulum, and so on. So it's a coordinated activity, and in that coordinated activity, if something goes wrong, it affects all the partners of that activity. So, if a drug is able to enter into the cytoplasm, definitely it is going to affect the mitochondria, the structure of the mitochondria, and the function of the mitochondria. We studied, we discussed three steps during last lectures, that how a xenobiotic can affect the mitochondria. It can affect the mitochondria by disturbing the membrane, first thing, by the affecting the citrate cycle, by the protonopheric activity, and there was another one, that is a drug may be treated as a pseudo substrate. A drug may be treated as a pseudo substrate. So in addition to that, the drug may affect the electron transport. In addition to that, a drug may affect the opening and closing of the mitochondrial permeability transition pore. These two steps we are going to talk today. Number one, how a drug or a chemical is responsible for the disturbance in the electron transport chain. So let us begin with this step, that how the chemicals they are affecting the electron transport chain. You must have heard about cyanide, potassium cyanide, hydrogen cyanide. These cyanides are deadly poisons. If a patient just put the cyanide on the tongue, immediately dies. Have you ever thought of it? Why it happens? It happens because of the blockade of the electron transport chain. The second example that we are going to discuss is doxorubicin. Doxorubicin is an anti-tumor drug. Doxorubicin is given to those patients who have developed the solid tumors, solid cancer. So doxorubicin or adriamycin, it is a patent drug, but this affects the mitochondria. So with these two examples, one is the hydrogen cyanide or potassium cyanide, in another one is the doxorubicin. We shall try to discuss the electron transport chain, disturbances in the electron transport chain. If you remember, in the what happens in electron transport chain, the important thing is the synthesis of ATP from ADP and PE, phosphate. And if there is something wrong with this activity, the electron transport is hampered. The electron transport is altered. And in that case, you find that the electron transport can be affected by the effect on the proton pump. We talked about the protonopheric activity. We talked about the that that chemical, the fungicide, the pentachlorophenol. The pentachlorophenol has a protonopheric activity. In the same way, the proton pump is disturbed, and then it is the inhibition of the ATP synthesis. Two steps. Then let us discuss what happens when cyanide is given. It binds to cyanide, inhibits the cellular respiration, the respiration of the erythrocytes. And how does it happen? It binds with the cytochrome. It binds with the cytochrome. And cytochrome, if you allow me to talk specifically, this is a cytochrome C oxidase, which is affected, which is bound. And you know that these bitter almonds, almonds, badam, हम लोग रोज खाते हैं. But if you take the bitter almonds, kadwa badam अगर आप खा लें, it has got 0.1% of potassium cyanide. So it contains a cyanide, and it may be poisonous. So this can result into hyperapnea, and it can result into red color of the skin. As soon as the potassium cyanide is taken up, it changes into the red color of the skin. And it is because of the inhibition of the iron containing enzymes, and this is again a family of enzymes. These are cytochrome C oxidases. And the second thing that happens is that it binds with the hemoglobin. Potassium cyanide binds with hemoglobin. Hemoglobin changes into methemoglobin, and then methemoglobin changes into methemoglobin formation can be treated with thiosulfate. So this is how if you just inhibit the electron transport, immediately what will happen? The cell will lead to death. The cell will lead to immediately all energy processes of the cell will be stopped, and cell will be leading to death. So one of the mechanism, why do we study mitochondria? We study mitochondria only to explain that how a mitochondrial injury is going to affect the cell, how it is going to kill the cell, how it is going to result into apoptosis, or how it is going to result into necrosis. This is what we are going to study through the study of mitochondria. So the mitochondria, one way of affecting the mitochondria is the potassium cyanide, which is affecting the iron containing enzymes, the cytochrome oxidases. And the second one that we are going to discuss is the effect of doxorubicin. I told you it is an anti-tumor drug. What doxorubicin does? Doxorubicin has been found to result into cardiomyopathy. That is, many people, when they are given doxorubicin, they die because of the cardiomyopathy. And that has been linked to mitochondrial injury. I tell you few steps that are involved in mitochondrial injury caused by doxorubicin. It can, doxorubicin binds with a molecule which we call as cardiolipin. And this cardiolipin is an electron acceptor. And there is electron acceptor complex in the mitochondria. We call it as we call it as quinine, and this quinine changes into semi-quinine, and then this leads to high redox potential, and it changes the molecular oxygen O2 into superoxide anion. It results into oxidative stress, it results into oxidative DNA damage, it results into the opening of the mitochondrial permeability transition pore. So doxorubicin, which is a drug used to treat cancer, may result into cell death through a mechanism, and that mechanism is central to quinone. That quinone changes, there is a quinone complex in the mitochondria, and that quinone complex changes into semi-quinone, and as soon as it changes into semi-quinone, it changes all the oxygen available there, molecular oxygen, change into superoxide anion. That is why to deal with superoxide anion, mitochondria has been equipped with superoxide dismutase. Right? The superoxide dismutase will take care of it. But if the cell or the mitochondria is overwhelmed by the superoxide anion, the superoxide dismutase will be inhibited, and the result will be the oxidative stress. And that oxidative stress will result into the damage to DNA, that will result into the opening of the physiological pore that we call as MPTP, mitochondrial permeability transition pore, right? So these two examples, they explain that the mitochondrial the mitochondrial function can be affected by all those drugs who are disturbing the electron transport mechanism, and that electron transport mechanism can be manifested through these steps which we discussed with the help of two examples. One is that of potassium cyanide, and the another example is that of doxorubicin. Now we come to the next mechanism, and that mechanism is this one, the central dogma of closing and opening of the mitochondrial permeability transition pore, MPTP. For that you must understand what MPTP is. MPTP है क्या? MPTP अगर आप कोई सेल बायोलॉजी की किताब पढ़ेंगे तो आपको माइटोकॉन्ड्रिया में कोई नहीं दिखाने वाला कि MPTP है क्या, ये क्या चीज़ है MPTP। उसका कहीं आपको कोई मेंशन भी नहीं मिलेगा कि कोई MPTP नाम की कोई चीज़ है। मैं आपको बताता हूँ ये MPTP है क्या, जिस पर सारा दारोमदार होगा। आगे हम जो डिस्कस करने वाले हैं, those two basic mechanisms of cell death, the apoptosis and necrosis. For that, it is very important for you to understand what this MPTP is. MPTP is a hypothetical physiological pore. कहाँ है? Where it is, this MPTP is situated, where it is located? It is located at the outer mitochondrial membrane, outer mitochondrial membrane. How does it open and close? It is a voltage gradient dependent phenomenon, that if the voltage or the electric current changes, electrical power or availability to the mitochondria changes, it results in the opening or the closing of the mitochondrial permeability transition pore. So now we should look at that there is the inner side of the mitochondria, there is the outer membrane of the mitochondria, and there are certain pores, and these pores are having some translocators, and these translocators are known as adenine nucleotide translocator, ANT. And in addition to that, it has got certain receptor molecules which can be received by this. So there is a complex, a protein complex. ये जो पोर है, ये है किस चीज़ का बना हुआ? ये एक प्रोटीन कॉम्प्लेक्स है, और ये वोल्टेज डिपेंडेंट मैकेनिज़्म है, जिसको हम कह देते हैं VDAC। अगर इसका फुल फॉर्म मैं आपको बताऊं तो वोल्टेज डिपेंडेंट एनायन चैनल, voltage dependent anion channel, VDAC. And this VDAC is responsible for the exchanger of ADP into ATP and ATP into the ADP, and there is something in the mitochondrial matrix, and that something is known as cyclophilin D, and there are others which are known as one is the pro-apoptotic factor, and another one is the anti-apoptotic factor. One is the pro-apoptotic factor is BOX, and the anti-apoptotic factor is BCL2. So a balance between BOX and BCL2 maintains or keeps the opening closed. Whenever a drug enters into the mitochondria, then what it does, it affects these mechanisms, and the result is that the mitochondrial permeability transition pore opens. And when it opens, it releases the cytochrome C. And when cytochrome C, you see, you might have heard about when we discussed about acetyl coenzyme A, we talked about oxalacetate, we talked about the citrate, we talked about the aconitase, we talked about all of those mechanisms which are responsible for maintaining the proper structure and function of the mitochondria. But now, what has happened that everything is going on very nicely, everything is being done very nicely, but what has happened that the drug has produced the oxidative stress, and that oxidative stress has resulted into the increase in the voltage, and the voltage dependent channel opens at say for example 1500D, and there are some reactive oxygen species formed, and these all you see release of the pro apoptotic factors occurs, and the cell enters into apoptosis, or the cell enters into necrosis. So this is how the opening and closing of the, we shall be discussing it when we discuss apoptosis again, and you will be able to understand it properly, that how this opening and closing is responsible for apoptosis. So what will happen when this pore will, mitochondrial permeability transition pore will open, it will release calcium into the cell, and that calcium will be responsible for the coagulative necrosis. Calcium is very important. Unfortunately, the man I had been talking to you, Sten Orenius, who belonged to Karolinska Institute, Sweden, he expired on 27th of April 2020, and he is no more with us, and we shall not be able to read much more interesting research that his group was conducting. So it is a sad news for biochemical toxicologist, that is Sten Orenius, expired on 27th of April. So whenever we talk of calcium, we cannot forget Sten Orenius. So what will happen, that one more thing I would like to discuss with you is that this mitochondrial toxicity has also been correlated with cholestasis. What is cholestasis? Cholestasis is you can understand it, the cholestasis is the formation of the bile, or you can just say it is the stone formation in the gallbladder. And that stone formation in the gallbladder happens due to disturbances in the bile and bile acids. So what happens is many of these drugs or chemicals, they like phalloidin. Phalloidin is a molecule coming out from phalloides, and phalloides is a mushroom. So Amentia phalloides. So this causes cholestasis, and this cholestasis is the result of the disturbances in MAP kinase. That is the mitogen activated receptor kinase, and the activation of this enzyme also, this has been correlated with oxidative stress, and oxidative stress is related to the apoptotic factors, and those apoptotic factors like BOX and BCL2, they are related with the cell death. So this cascade of the mitochondrial injury, which we summarized into 5 basic mechanisms. Now you can just summarize it, what those 5 basic mechanisms were. Those 5 I will be sending you a very good figure dealing with it. Inhibition of citrate cycle, protonophoric activity, accumulation in the matrix of small xenobiotics, then inhibition of beta fatty acid oxidation, inhibition of electron transport, we just discussed, inhibition of ATP synthesis, that is inhibition of ATP synthetase, we just discussed, release of cytochrome C through the MPTP opening, and the binding of xenobiotics to cardiolipin. We discussed this just now. So these are some of the possible mechanisms of mitochondrial toxicity caused by different xenobiotics. So with this, we finish our discussion on mitochondria, effect of xenobiotics on mitochondria. And now we shall be heading towards, first we shall discuss apoptosis, and then we shall discuss necrosis. I hope you will enjoy this.